Thanks, Arno Pinaki, for inviting me. It's a great pleasure to be here. This is my first time at IMSC Chennai. The campus looks beautiful, very quaint. Great to be here. Just very briefly, I'm at the Center for Nanoscience and Engineering at IISc Bangalore and also associated with the Department of Physics. I also have involved in a startup that we had co-founded, and I'm going to tell a little bit about how self-propelled magnetic nanomotors have resulted in a medical device startup. Also, I realized from the audience that it's very diverse, and in our group, we sort of have two vertical threads. I'm not going to talk about our experiments on quantum fluids, where the question we are trying to address is, how does a two-dimensional solid melt at 0 temperatures? But rather, I'm going to talk entirely on nanomotors or nanoswimmers. And these are the sort of questions that we are addressing. How do you make a model active matter? And how that can lead to some form of a new therapeutic strategy for medicine. Before we start, let me acknowledge the large number of collaborators from multiple different disciplines who have contributed to this work. And definitely a large number of students, very talented students and postdocs who have done this work over the years. To tell you the basic premise of our experimental system, we are talking about a wet active matter. We are talking about a colloidal particle, a very small particle moving in a fluid. The Reynolds number is significantly less than 1. So in other words, the motion is entirely dominated by friction. This is, of course, the world in which microorganisms swim. And just to remind you of Purcell's paper, the typical motion that is used are either the corkscrew motion of an E. coli bacterium or the way a spermatozoa swims, which is a long, flexible filament that beats and moves forward. Of course, this is not the only way to move at low Reynolds numbers. One can also imagine some form of a phoretic strategy in which you have an asymmetric structure, and through either chemical reactions or light or electric field, there is some form of a gradient of some molecules to the left and right of it, and as a result, there is a phoretic velocity that one would achieve. I'm going to focus entirely onto this line of work, just to tell you a little bit about the type of applications that people imagine would happen here. It's of course all started with Richard Feynman's very famous lecture called Plenty of Room at the Bottom, and there he talks about how these things, little machines, he calls it miniaturized surgeons that can be put inside human blood vessels and going and delivering drugs and treating diseases and things like that. If you were to make a research program out of it, the type of questions that you want to address is, firstly, how do you move very small objects in fluids and gels? You, of course, want to do it with good control from a distance, and you want to really fabricate and maneuver a swarm. You want to give it multiple functions so that they can do the sort of things he talks about. Interestingly, because we have a swarm, there can be interesting collective behavior, and that can, of course, result in some form of intelligence or tasks that single swimmers or single motors cannot do. And the type of applications can really be, there's no limit there. Now, one first question that one needs to address is, how do you power these motors in order to make them move in fluidic environments? And in this respect, magnetic manipulation is extremely well suited because it's fuel free, it's very, very biocompatible. Because it's remote, it can work deep inside an organ. And I want to maybe stress upon this point a little bit and also tell you what I do not mean by magnetic manipulation. So this is not a magnetic nanomotor in the sense I want to use it. So here you have a little Eppendorf tube with some magnetic particles. If you hold a magnet nearby, of course, all the particles are going to go near the magnet. Now, if you want to look into it a little closely, this happens because there is a strong gradient field, and this particular strategy will not work once you take the magnet a little bit far away or if you make the particle size smaller. It's just the way the force depends on the magnetic field. So one of the main motivations or constraints here is that you want to use some form of a homogeneous magnetic field. There's a very nice paper from the ETH Zurich group about how should such systems work. The strategy that we have adopted in our group is to mimic the motion of a corkscrew, and what we have basically is a artificial screw with a shown in green here. The red is the magnetic moment. In the presence of a rotating magnetic field shown in blue, the screw rotates and therefore translates. And this is really sort of the version 0 of our experimental system. But I'm going to build upon this a little bit. The head here is just something that comes due to our fabrication once in a while. We can avoid it. It's not a necessary aspect of this experimental system. So therefore, there are certain differences between this and a typical E. coli that is swimming. There is no counter-rotating head. There is no flexibility in the filaments, so on and so forth. So we are talking about rigid corkscrews sort of going through, propelling through a liquid. That's what we are talking about. The experimental system, let me tell you how we make these tiny corkscrews. This is made using a technique called glancing angle deposition. The idea is you have a substrate at a very extreme angle with respect to some material that you are evaporating, that you are melting. And this angle alpha is of the order of 85 to 86 degrees or so. At this extreme angle, the material that comes will go and stick to the substrate, but then any material that comes subsequently will get shadowed because of the part that has already been evaporated. So what you use here is to utilize this shadowing to make structures of different shapes and sizes. So if you do not do anything, there is just going to be some columns that are going to get grown on this substrate. But then if you start rotating this substrate, you can actually control the direction in which the columns grow, and simply by controlling the rate of rotation and the evaporation rate, you can make structures of different shapes and symmetries. So this is, for example, a helix that's grown, and well, this is very often what we have made in the lab. And just to stress upon the versatility of the technique, you are actually talking about a wafer scale system of small helices. So we are making 1 billion helices in one go, and as opposed to many other experimental techniques which are somewhat more limited in yield. So this really allows you to go and deep into sort of looking at collective properties and stuff like that. So just to also stress upon the versatility or the the range of parameters that one can get in the experiments, we are talking about swimmers. This is one of the largest, a couple of microns long, almost a micron width. And in opposed to this particular swimmer is one of the smallest in the lab, and here the scale bar is 500 nanometers. So we are really talking about more than an order of magnitude in what you can achieve in your experiments. Typically, these objects are made of silicon dioxide, which is just very easy to grow, and then we include some material, iron for example, into it, and all these experiments are done in multiple coils of our lab, depending on what type of applications we are talking about. So it's a triaxial Helmholtz coil, which means you can produce time-varying magnetic fields in any direction, any polarization, and also observe it simultaneously in microscope. This guy is a little bit bigger because we also put some animals out here for some of our biomedical experiments. But then very often we are able to see this with a really reasonably good precision. The type of motion one can achieve is definitely beats Brownian motion. Simply by changing the sense of rotation of the field, you can make the swimmers go north and south, or if you want to do east and west. And if you want to get some publicity, you can try to write some G-clef sign, some music sign. And of course, you're not just limited with one swimmer, but you can actually have a swarm of swimmers all actuated in a nice manner. Yeah, so effectively, it's the direction of motion is always perpendicular to the plane in which the field rotates.
But the orientation there?
No, the orientation is set by the plane. So you can, and I'm going to come to this in just a little bit. That's what the trick is. Okay. So yeah, so that means if you have lots of them, they are all going in the same direction. And that's something that's important. Yeah, but we will break that. Right. I'll go a little bit. So I wanted to mention, so you have a swarm, and then starting from this paper about 15 years ago, there has been actually multiple tech transfers on this, and there are people who are trying to put this in brain, in teeth, and a couple of different places. So in fact, there are some clinical trials that are happening right now with this particular technology. Initially, I think starting from our first work, one of the main motivation was to see how these things move in different biological fluids. And in fact, over the years, it's kind of funny, we sort of starting with water. Of course, there is teeth. You're actually observing this motion in human teeth. You can also move these things inside living cells, in blood. This is not in vivo, but ex vivo. This is outside, taken outside. This is a great tool to study biophysics of cancer. This is motion in cancer extracellular matrices, and this is something new in which you're barely seeing it, but we can now observe the motion of a swarm of such swimmers inside live animals taken with in vivo imaging. So we are actually very close to the fantastic voyage dream in some sense. So this is the peritoneal cavity, which is sort of a fluid-filled cavity in the gut, and is connected to many organs. The experimental system, you can have multiple materials and functions, so which really allows you to put in lots of different functionalities in one go. And so, for example, here the iron is placed in three different islands. I'll go a little bit quickly because I saw some optics-related talks. You can actually integrate silver nanoparticles onto the swimmers. So now what you have is you can have plasmonics integrated with magnetic swimming, and one of the aspects of plasmonics is when you shine light, it has a very strong light-matter interaction, so it works as little optical traps. So this is a fun experiment that we did some time back in which we took the swimmers. They have these plasmonic properties. You shine light next to some colloidal particle, so of course it works like a little trap, and then you can of course take it from point A to point B, turn the light off. So what you can now do is to sort of have trap, transport, to release little colloidal particles with very, very low light intensity. And the light intensities that are used here are orders of magnitude smaller compared to a standard optical tweezer. So that's the sort of things we need to, you can use, you can combine it with other fabrication schemes. I have been extremely lucky to be located at the Nano Center at IISc, where all these facilities are available, so which really makes life very, very easy. And of course, you can also do standard chemistry in which you can functionalize things to this helical swimmer and give it new types of properties. I'll go right into the business. So over the years, you could imagine that we have studied this motion in reasonably good detail. There are some caveats that we must mention. So of course, this is the propelling motion that I keep talking about. But if you think about it, you see, this particular motion is equally possible, right? You can have either a propelling as well as tumbling motion in both the cases. The magnetic moment rotates in sync with the external field. So, of course, the question is what happens, and one can actually write down and one can observe this type of tumbling motion as well as this type of propelling motion and motions in between. So these sort of things can be studied, can be calculated. Yes.
Can't you stop the tumbling? Can't you stop the tumbling by using another constant field?
Right. So then it becomes some form of a precisional magnetic field effectively, and you can also calculate the dynamics under such precisional fields. Right. So typically what happens is when it tumbles, it actually is again very close to the surface, so it sort of walks through, sort of a thing. I guess the key point is, there are lots of interesting, one can look into the details of this type of dynamics, look at the, I don't know how the geometry, the surface effects, the magnetization strength, so on and so forth. I want to highlight, and I'm not going to talk about this, that this is really a great system to also make local rheological measurements, and I have some work, we can discuss it some other time, where we can actually make little rheometers for even to make rheological properties of inside living cells. Okay, so that's the type of things that has been done and in process. The focus of this talk is, I said all these things because, okay, I wanted to sort of describe the experimental system in some detail, and I'm going to basically start from that question that was asked, that, you know, if you have a helical swimmers and if you have a rotating magnetic field, all of them move in the same direction, and this is of course not the property of a self-propelled system of swimmers, right? They're not self-propelled, they're just driven. And why do we want to do that? Well, of course, what we have is, I would call it still a propeller, which is externally powered, but more importantly, the direction of motion is determined experimentally, and this will be a zero force but a non-zero torque system if we use the typical language that is being used. On the other hand, if you take standard biological swimmers or even some of these catalytically powered nanoparticles, you will see that, well, the source of energy could be chemical or external, that doesn't matter, but crucially it's a zero force, zero torque system in which the direction of motion is not governed experimentally, but only governed by either orientational diffusion or governed by interactions. So the question that we wanted to address is, well, can we make an all magnetic active matter where the fuel will come from small magnetic fields, as we have been doing in our experiments. However, we do not specify which direction they should go, so their direction should only be governed by interactions with each other and the surrounding, similar to the chemical motors. That's the problem that we wanted to solve, and the motivation of this is, of course, to see what happens to such swarms, and we have just started to do this now. But let me tell you how one can have an all-magnetic, a self-propelled particle in which the fuel is small magnetic field and the direction is not given. And the answer is extremely simple. All that you have to do is you have to, this is the helix, and then of course if you have a rotating field, they're just going to go in one direction. But imagine instead of a rotating field, you give a simple oscillating field, and in other words, you only have Bz, which is perpendicular to the plane at which the object is residing, and then the moment is going to point upward, and we are just looking at it from the end of the helix, and then when you go to plus z to minus z, the helix would have to turn, right? The magnetic moment would have to point downward, and when it does that, it can, the helix can go through either a clockwise turn or a counterclockwise turn. So you can do a clockwise or you can rotate in the counterclockwise turn, and because the helix is a chiral object, the clockwise turn can result in maybe a forward motion, the counterclockwise turn will result in a backward motion. So whenever you go from a plus Bz to minus Bz, is going to have a little bit of a motion, and when you go it back, when you again get the field up, so you're just oscillating the field, again there will be either a clockwise or a counterclockwise motion to go back up. Now, the key point here is, at no point of time are you telling the helix which way it should orient. Okay, that's the key point, and that's what you are seeing in this movie. So these things, when you do the experiment, you find out that, well, there is not an equal probability to go from, to whether it should do clockwise or counterclockwise, where it goes from this state to the plus Bz to the minus Bz state, and then similarly, there is a specific way in which it goes from the minus Bz to the plus Bz configuration. And this simply happens because there is some natural weight asymmetry in the system which makes it always turn in one way, and therefore it goes forward, and then when you turn the field back towards plus Z, it comes back using a backward motion. So really what we did, and this is V1, is some kind of a magnetic reciprocal swimmer, if you use Purcell's language. Yes, Mahesh?
Then Purcell's language, Scanlon's theorem would require that there is no symmetry breaking, therefore it will come back.
And that's exactly what we are doing here. It is what we have is a scallop.
I was under the impression you were trying to avoid that.
Yes, I will avoid it. Just in one more slide. So what we have is a reciprocal swimmer. and this reciprocal swimmer is simply going back and forth. But we do observe an enhanced diffusion even for a reciprocal swimmer, and this is actually, so this is Brownian motion without doing any magnetic field, and now it's just going back and forth. It shows some form of enhanced diffusivity, and this is not very surprising because as it goes back and forth, it's also doing some form of an orientational diffusion in its plane. So in other words, when it goes, it can diffuse and then it can come back a little bit. So effectively what you have is a rotational diffusion. So what we have is, it is an active matter, it's still boring, it's still a reciprocal swimmer, right? So that's what we have here. So of course the next question that one should ask is, well, can you break the reciprocal symmetry? And just to give a little bit of details of how this whole thing is working, you do have the plus Bz here, this is your magnetic moment, this is a little bit of a weight asymmetry, which says that when you go from plus Bz to minus Bz, it should take a clockwise turn to go back, right? And then when you go back up, it should go back by a counterclockwise turn. The trick is really very, very simple. All that you should do is when you make it go from minus Bz to plus Bz, you should just, and when you do this, well, about 100% of the times it's just going to do a clockwise turn and therefore go forward. The trick here is you just need to go a little bit faster when you go from the minus Bz to the plus Bz term. And when you do that, it's basically, and you can show it with all the math, we did it in our papers, that there is going to be an equal probability to take either the counterclockwise or the clockwise paths to make it point towards plus Bz. So effectively what you have is, it's not really nothing but a ratchet that is being used. And this of course only works in the presence of thermal fluctuations. The key point is, after one such cycle, you have some preference towards clockwise turns, at least in the picture that we have shown here. So of course, if there is no thermal fluctuations, this is simply going to be back and forth. So the noise is indeed necessary to break this counterclockwise clockwise symmetry. So what we are really using is the Brownian ratchet to move things forward. These are some experimental results. So, you know, if you can do slow and fast switching, you can actually go from, when the slow switching is perfectly deterministic, there is this fast switching which gives some range of stochasticity. And the interesting thing is, you can actually compare simply by varying this ratio of the times that we use in the experiments. You can actually go from a fully reciprocal to a non-reciprocal swimmer. So you can actually control the degree with which you are able to control this. This was the work. So, and really what effectively we are doing some form of a ratchet out here, okay? So that's the way in which you can make it go from driven to a self-propelled swimmer. And the next that we want to address is, what are the differences? If you take a swarm of such swimmers, what are the differences? We have a driven versus a self-propelled system. In order to do that, I'll start from here. Well, maybe this is the time when one should ask, well, how do the swimmers interact with each other? At very short distances, they of course interact magnetically, but at the typical distances that we have in the experiments, hydrodynamics is far stronger and far more long-ranged. So one of the things that we did is to actually look at the hydrodynamic flow that the helix produces as it swims. So this is a work in collaboration with Alex from Technion, and what we did was we took the swimmers and we did not consider the swimming in bulk medium, but we did assume things to be reasonably close to a surface. And I don't have a pretty picture, but effectively what happens is when the swimmer swims and it creates this rotlets, because of the presence of the surface down there, effectively what you have is a very strong lateral flow. So this is an interesting swimmer in which if the swimmer is going from left to right, most of the flow is actually going from north to south. It sort of drags things, and in fact and that has some consequence with which the swimmers actually interact with each other. We do understand these swimmers, the hydrodynamics reasonably well, and to a large extent you can completely ignored the fact that it's also moving in this direction, because it's really the sideways flow that just dominates things a lot. So there are this long-range, non-reciprocal hydrodynamic interactions. I say non-reciprocal because if you take two swimmers that are parallel to each other, you will see that my left hand is pushing my right hand away, and my right hand is actually pulling the left towards it. So there is some form of a non-reciprocal hydrodynamic interaction between the swimmers, which actually results in, which is kind of interesting when we go to the active case. So back to the driven system, where we simply have a rotating field. I'll first show you the boring results. So this is, of course, when a lot of these things are all going in the same direction, and they are really not interacting with each other because, well, it's a low frequency, low density, they are moving very slowly. Things become interesting, a little bit more interesting, when you have a large number of swimmers that are actually, we wanted to drive it from left to right, but what you would see is a very strong drift towards the right, and that's really because they are all sort of, as they try to go in some direction, they're also making them drift in a sideways because of the hydrodynamic flow. And the interesting thing about this is, and this is the part that I like, if you really analyze it very, very carefully, you would see that the sideways drift depends strongly on density. So now what you have is an interesting handle with which you can actually have swarm control, and you can look at the response of the swarm and connect it towards the local density, and that actually is could lead to interesting ways in which you can control the swarm. We are just in the process of finishing this. Things get far more interesting when, instead of a rotating field where everything is driven, when you start looking at how things happen in oscillating fields when everything is active. And in this particular case, I mean, of course, this is really some form of a strong, dense collection of active particles. We have taken a few particles and plotted their trajectory, and of course we start seeing this type of swirling motions, and this is sort of prototypical active matter, wet active matter with strong hydrodynamic interactions. We do not see MIPS or any phenomena like this. We are just starting to look into this very, very carefully. Sriram is trying to build some form of a field theoretical model out of this. So there is a lot of work in progress. There are some funny things we observe, and I'm just going to show you this. Well, firstly, for no reason, this particular little defect, some big, some stuff that's not even magnetic, started moving in one direction. Can you see this very closely? And if you can observe the dots here, and this is kind of interesting, you will see that they will all come together. They're accumulated, and then suddenly they're all going to go all at the same time. So I mean, you have to look at this part. We really tried to make the movie nicer, but I can maybe show it to you later. So they're all here, all here, all here, all here, and boom, now they all go away together. We don't understand what's happening, and this is really the reason why we need some serious theory to understand this better. But a lot of effort went into building this experimental system, and just to give you an idea, I mean, of course, this is just some toy model, and these are just some of the little hydrodynamics toy model sort of stuff we did in the lab. Again, we don't understand this very well, but it does seem like some strange behavior. I'm going to just say that, well, the typical diffusion constants that you can get out of these active particles is almost, more than 50 micrometers square per second. Just to put things in perspective, this is almost 300 times compared to what is there in the passive case. So they are really very, very strongly active, very high Peclet number, and I'm just going to say that this is work in progress. Okay, so hopefully we'll be able to report you something better in some time. I have another. Sorry? Okay. Thanks. How does this result in a new therapeutic strategy? And this is a question one can ask for everybody. One of the possible ways in which active matter can be used for new type of therapeutics. So one of the key points to note here is, well, because the mean square displacement is higher, self-propelled objects can move in all directions, they can go in different ways, and they can cover more areas in less time. And that's one of the benefits of having active matter. And one of the places where this really becomes very, very useful is actually the inside of the tooth. So I don't know how many of you had root canal procedures out here, but let me remind you a little bit. In root canal, this is the structure of the tooth, the part in white, and what you have here is the pulp. And typically in a root canal, the dentist comes, takes the pulp out, and they put some gutta parka or some sort of a thing to just seal it off for good. So the tooth is not alive or not viable anymore. So the pulp is really where the bacterial infection happens. That's where the decay happens. But if I were to go a little bit into the structure of the tooth, this is the so-called dentin tissue. The structure of dentin, here this is the pulp, this is the tooth. Dentin, this tissue, is made of a large number of tiny canals. And these canals are about a micron or so long in diameter and about a millimeter or so in length. So if I were to sort of say it in a very simple term, what we have is a cylinder. You can imagine the tooth being concentric cylinders, and they're being joined by little canals that are going radially outward. Now, one of the biggest problems why 10% of root canals fail is because of the bacteria that are sitting out here, which the doctor is not able to remove during your root canal. Typically what they do is they put sodium hypochlorite, which is bleach, and they hope that the bleach is going to diffuse through the dentinal tubules, go all the way up to a millimeter and kill the bacteria. And because it's bleach, this typically doesn't happen because the time it takes for the green one, this is typically the diffusion constant of bleach, is probably going to be, it's to go all the way up to a millimeter or so will take many, many hours. So of course, this is one place where active particles are very useful, where you can go in all directions and cover a lot of region, go all the way up to a millimeter very easily, and something like 8 microns a second is actually quite good, because you are able to go to the depth of the tooth within about a couple of minutes or so. There are lots of companies and lots of technologies who are actually trying to solve this particular problem, because 10% of the number of root canals, just imagine, we all have, 28 teeth, and just if you do the numbers, it's just it's really staggering. But none of these other techniques are able to go deep. So of course, this is what we did. We put the swimmers, and what you are seeing here in this movie is actually motion through the dentinal tubules. We can actually drive them because these things are really, really tiny. And this is one part where driven versus self-propelled makes a huge difference. For the self-propelled ones, after some 20 minutes or so, you can go only in one of the directions. So this is the central pulp region, and this is the dentinal tissues. Of course, you did not move much over a certain time. On the other hand, with self-propelled, we could actually cover the entire region in a much shorter amount of time. Just to tell you a little bit of application, so you can actually make them actively diffuse in all direction, cover all of the tooth, and then you can use a rotating field and even retrieve them one after another. So that's again one of the benefits of having a system that can be both active and driven at the same time, simply by turning a magnetic field knob. In this picture, what you are seeing is here these are swimmers, the green points. Here you are looking at the green points correspond to living bacteria, and the red points correspond to dead bacteria. May not be very useful here, but the key point is you can take the swimmers and actually kill the bacteria in the location that you would want. I'm going to end my talk here and just say that, well, you can compare the key difference between some form of active propulsion versus some form of a rotating field, and you would be able to actually see a dramatic improvement when you have self-propelled objects moving in all directions. And this is again partially the reason, because here topography is making a huge difference. So what we have here is things from the central region of the pulp are going through the tubules in all possible direction. You are not allowing them to reorient. So effectively, if you think about the active Brownian particle model, you are not allowing them to reorient. So as a result, the overall displacement that you can achieve in a certain amount of time is significantly more because of the topography. So active particles under confinement with reduced orientation can really play a very important role in covering a large region in a small amount of time. With that, I would like to conclude the key point that I spoke about today is you can make the swimmers multifunctional, and this is it's work in progress of how we are studying making this as a model system to study active matter, and there is some relevance to practical medical applications. The part that I did not tell you about today is this is also a fantastic new tool for intra and extracellular mechanical measurements. With that, I would like to thank you for your attention.
So you said that the reciprocal swimmers are kind of boring, but if you put many of them together, the many body effects are not boring. They will not drift, the reciprocal swimmers, but if you have two of them, they'll still rotate. The air will rotate. If you have a strongly confined geometry and you have a reciprocal pneumatic swimmer, a pair of them can rotate. A single one cannot.
Yeah, indeed. There are these interesting fun papers from, I think, Eric Logger about no many-scallop theorem sort of a thing. So you can have two reciprocal swimmers. If you have the right phase differences, they will behave like a non-reciprocal system. So.
Yes, but more importantly, the many body effects can give you a sort of a state which is aligned but spontaneously rotating.
Okay.
Things of that sort can happen.
Yeah, so I don't know if that will happen if you confine it in 2D.
Yeah, this is when it's confined.
Oh, is that so? Okay.
This is, I mean, it cannot happen in 3D.
Okay.
It can happen in a 2D interface, or it can happen in
But you are talking about which type of rotation? You are talking about
Yes. So this is the long axis.
Right.
Rotation. So if you have two of these objects.
Okay. Yes. Absolutely.
This is a subjective query, so there's another school of thought which claims that the Janus particles are also extremely helpful in drug delivery and stuff like that, because of the targeted motion and self-propulsion. Would you would like to compare what the pros and the cons between the two strategies?
Well, I suppose one of the benefits of Janus particles is you can probably build in chemotaxis and rheotaxis, probably chemotaxis very easily. And there is this interesting work from the Barcelona group who's using this swimmers to, I think, target some cancer of the urinary bladder.
Sergei's group. Yeah.
Right. So Samuel's group is doing this, and what he has done is he uses urea as the fuel, and then he's able to actually penetrate deep inside the cancerous tumor. And here again, I think chemotaxis is one place where the Janus motors make a very strong case. So.
I have a question about this retrieving this swimmers using this rotating field, right? When you retrieve from this dental this thing. So when you put this rotating field, if it retrieves from one side, doesn't it push the other swimmers this thing? So how do you retrieve everything together?
Yeah, so you have to create a flow, and over a time you effectively can collect. Sure, so some of them will indeed go in the other direction, you're absolutely right. But the whole thing is, you can imagine there is a wall and there are a few holes, so most of them are just going and banging their head against the wall. So. No, so this comes from the natural topography of the dentinal tubules, but we have also checked it in other types of sort of gels. So in gel-like media, you would actually see that the reorientation time is significantly longer. So as a result, it sort of moves effectively, it follows t-square rather than t-dependence for a longer time.
Okay, so it sees the boundary and then it doesn't.
It does not orient. So it just, if there are two of them, they will continue going in this manner for a longer time. And this actually is very dramatic. These are orders of magnitude improvement.
I had another question. So maybe I just didn't follow. You had an expression for the MST?
I think it's incorrect.
Yes.
I think I missed a term. I just noticed it a little bit ago. There is some problem with that expression.
Which is the.
No,
the
I think.
V-naught is.
V-naught is the active velocity, but I think I missed a term
Which
there.
is emanating from the magnetic field?
Yes. how do I go there? Oh, so V naught is the active velocity, which is the effective active velocity that you would get. I'll just go quickly there. Sorry, 1 second. Yeah. So I think
Which you will calculate from the,
that's the self-propulsion velocity. So we just did the fitting of this mean square displacement, and this is in turn a function of the fast and slow time scales that we put in the experiment. So you can control that. And tau r is just the rotational diffusivity of the swimmer.
thank you.
in plane.
I have few questions related to the experiments. So, what happens near the boundary when this particle goes to the boundary?
So they are already close to the wall, and if they are, and if the interaction is too strong, which depends on sort of the chemical properties, they will not only go forward but also have a strong sideways motion, even in isolation, even a single swimmer. Typically, we make them charge so that they are somewhat farther away from the surface, so they move in one direction.
So they don't follow the boundary when they reach near the boundary.
So they would have a sideways motion if they are very close to the boundary. And I'm talking about the one that matters the most is if the swimmer is going in this direction, how far is it from the bottom surface? Typically.
And that PIV that you showed, so
Sorry.
Oh,
yes. So these PIV are for the hydrodynamics that is happening at the background. So these are background flows that are created by the particles.
By the swimmer, yes.
And you trace that by some tracers.
Yeah, we have done PTV, we have done PIV, they all match. pretty
So do you do this PIV by putting the tracer particles or?
So we do PTV. So we have also done, yeah, so indeed. So we have put tracer particles, we have calculated, measured their trajectories, and from there we are able to get this flow.
Can I ask how small is your tracer
So that
particle?
depends on the, yeah, so that's a good experimental challenge. It depends on the viscosity. We typically stay between 200 to 500 nanometer particles. It depends on the viscosity.
And won't this tracer particle change the dynamics?
Yeah, so we don't think so because the densities are very low. This is really averaged over a very, very long time.
One last question.
Thanks.